folks, and welcome to this microbiology video where we're going to show you how to perform a gram stain, a differential staining technique that allows you to see the difference between a gram positive and a gram negative, which is ultimately indicative of the cell wall outside of the bacterial cell. Okay, so we're going to begin by getting our Bunsen burner going. And get your slide ready. We already have a slide set out, and you want to label your slide with one of these wax pencils. I'm labeling it gram because we are simply performing a gram stain. There's not much room on these slides to put a label, so you need to get familiar with a shorthand that you can recognize once you start working with multiple slides. So we have our slide labeled. Next thing we're going to do is get our loop sterilized and ready. Now while this is cooling, I'm going to go ahead and point out that the beginning of the gram stain is very much similar to the simple stain method that we've already shown you. And in that method, we began by creating a smear across the slide. Now, if you are collecting your bacteria from an agar plate, you will want to make sure you use some sterile water to make that smear. But today in this video, we're simply using a broth culture, so we don't need to use the sterile water to create that smear. Once this is cool, we can now get into our bacteria. We want to make sure it's mixed well enough, so give it a few flicks. Using sterile technique, access, tube, we have bacteria now on our loop, and we are simply going to spread it around just like we did for the simple stain. And now we're going to sterilize and let that air dry. Now while we're letting that air dry, we're going to talk about some of the principles behind the gram stain. You see here that we have several things lined up, and this is what you're going to want to have lined up, just like this. You're going to be working with your partner, so make sure that you aren't trying to start your gram stain while they're using some of these materials and vice versa, because this is going to go one right after the other. Especially when you get to this step with the alcohol and water, you're going to need to have that ready and on hand. No delay in between. So the first step of this process is crystal violet. And it's a simple stain with crystal violet. Once we heat fix this, we'll add stain to the top of it and let it sit there for a minute, and then we'll wash it off like we've done with simple stains before. But then we're going to follow that up with iodine. Okay? And iodine is going to lock that crystal violet dye into and in between all the NAG and NAM particles of the peptidoglycan in the thick cell walls of the gram-positive cells. Okay? Now, once we finish with the iodine and we've locked up all the crystal violet, we're then going to wash with ethanol. This ethanol will ultimately clear away any of the lipids in that outer membrane of gram-negative cells, and because gram-negative cells have a very thin peptidoglycan layer, there's hardly any crystal violet that's been locked away once the iodine has touched those cells. And so the ethanol ultimately washes away any crystal violet from gram-negative cells. Because the peptidoglycan is so thick on gram-positive cells, the crystal violet gets locked in and is not washed away. However, as we'll point out, you're only going to wash for 3 to 5 seconds with the ethanol, because if you wash long enough, you can eventually wash the crystal violet out of your gram-positive cells, and then the stain is a wash, literally. Okay? So, once you clear the crystal violet off your gram-negative cells, then you're going to come in with safranin. Safranin is a counter stain that will then allow you to see the gram-negative cells that have had all the crystal violet removed from them. Safranin is a red dye, crystal violet's a purple dye, so your gram-positive cells will appear purple, your gram-negative cells will appear red. this point, our slide has air dried. We're going to do a quick heat fix. Again, 5 passes over the flame. 1, 2, 3, 4, 5. And if you're worried about burning yourself, make sure you use a slide holder. Okay. So with this heat fix, now we are good to start the simple stain process. So here's your crystal violet. And again, if you're concerned about getting dye on your fingers, make sure you are using gloves. So get a little bit of dye. Put it right over the point where we made our smear. And again, you don't want to be bashful with the dye. You want to make sure that smear is completely covered, but at the same time, don't be wasteful. Set a 1-minute timer and wait. All right. So while we're waiting, since I just gave you a lot of material, let's review again what this crystal violet is doing. The crystal violet is a dye that is soaking in between all of the NAG and NAM particles of the peptidoglycan in the cell walls of bacteria. So what this is doing is that purple dye is getting in between those particles, and on a gram-positive cell, that layer is super thick, so there's a lot of dye getting in and staining those gram-positive cells, all right? Now, one thing that you'll notice different from what you see in your book is that your book suggests you only need 30 seconds. What we found is that some of our cells that we work with, their cell walls don't soak up the dye nearly as well. So you need to let it sit for at least a minute, okay? So that's what the crystal violet's doing. Here's our timer. And we are simply going to rinse off, just like we did with the simple stain. Here's your water. And don't spray it right onto the smear, up and over, and tilt back and forth until you get a clear wash. All right. Set this back down, and then come in with iodine. And again, place the iodine directly over where you have your bacterial smear. And same with the dye, don't be shy. Now, the iodine in this process is what we call a mordant. A mordant is a chemical that binds with others to lock it into place. In this case, we're locking our crystal violet dye into that peptidoglycan cell wall. Okay? Got to set my timer, there we go. So again, we're waiting a minute. So our iodine is locking crystal violet into the cell walls of our gram-positive cells, whereas our gram-negative cells, there's some peptidoglycan there. We've talked about that. It's there, but it's super thin in comparison. So yes, the iodine is locking crystal violet into those cell walls, but there's not much being locked in. So in the next step, when we come in with ethanol for 3 to 5 seconds, all of the lipid covering that outer membrane of the gram-negative cells is going to be washed away, exposing the very thin peptidoglycan underneath, and then all of the crystal violet is going to be washed away from there. Okay? Oh, there's our timer. And now we are going to rinse away 3 to 5 seconds with ethanol. And then we are going to immediately wash with water. The reason for that is, like I mentioned, the ethanol will get rid of the crystal violet. And even though crystal violet is locked in strong to the gram-positive cells, because that cell wall is so thick, that ethanol can eventually permeate and get rid of the crystal violet within it. Okay? So we want to make sure that ethanol is gone and not staying on the cells, and slowly washing the crystal violet out. Once we've done that, we then come in with safranin, basically another simple stain. Make sure it's right over where you have your bacterial smear. Don't be bashful, make sure it's covered. Set your 1-minute timer. And what this is doing now is, once we washed our cells with the ethanol, we cleared out all the crystal violet from the gram-negative cells. So now the gram-negative cells, if we were to put the slide under the scope at that point, they would be clear. You wouldn't be able to see them. The only things you would see are the purple dots indicating gram-positive cells, because those are the ones that were still holding on to the crystal violet dye. However, once the gram-negative cells are made clear by the ethanol wash, we can come back in and counterstain with this red safranin dye. The gram-negative cells, which have no dye on them, will now readily soak up the safranin dye and hold on to that red color. Okay? So, simple dye with crystal violet first, mordant to lock in that crystal violet with iodine, ethanol wash to get the crystal violet out of the gram-negative cells, wash off the ethanol, and counterstain with the safranin for the gram-negative cells, which we are now going to wash off with just some water. Again, turning it back and forth, make sure that we get a clean wash. At this point, got a slide ready. And to finalize this process, get out some bibulous paper. Blot dry. Now you have a slide ready. However, we're not done at this point. We want to make sure that we get this mess cleaned up. So the first thing first, make sure, just in case, everything is sterile. Go ahead and make sure this is clean and ready to go for the next person. And then you have your tray here with all the stuff we washed off. First thing, we're done with the gas now, so we can turn off the gas. Turn on the sink, let it run for a few seconds, and then simply pour the contents of your dye tray into the sink, and then rinse the tray itself. Let it drip dry for a moment over the sink, and then set it on the side of your lab bench to dry. Let the sink run for a little bit after, since there's dye there. We will want to make sure that is diluted out sufficiently before it enters the system. And after about a minute, you can turn that off. This is ultimately how you perform a gram stain to visualize the difference between the different cell walls that you can find on bacteria.